Hello my dear students, how are you? Hope you all are doing good. So today in this video we are going to discuss about the various types of cell organelles and their functions. As you already know that a cell has different types of functional unit which are known as cell organelles. So in this video we are going to discuss about the various types of cell organelles just like cytoplasm, chloroplast, endoplasmic reticulum. We will discuss about the nucleus. We will discuss about the cell wall. We will discuss the function of cell membrane, Golgi apparatus. We will discuss about the vesicles, vacuoles, mitochondria, lysosomes and many more. So are you excited? Let's begin in this chapter. So before going into depth of cell organelle, I would like to define the term cell organelles. A small organ-like structure present inside the cell is called cell organelle. It has a particular structural makeup and performs a specific function for the cell. So on this screen, you can see the different types of cell organelles individually. Okay, here it is a nucleus, endoplasmic reticulum, Golgi apparatus here, then cytoskeleton, vacuole, we have cell membrane here separately, the structure of ribosome is shown here, lysosome, centrosome and in the end mitochondrion. Okay, so now we are going to deal or we are going to learn separately about these cell organelles one by one. But before going into depth of these cell organelles, I would like to tell you that depending upon the presence or absence of membrane, cell organelles can be classified into three categories. What are these categories? These are organelles without membrane, and the second category is single membrane bound organelles. Third category is double membrane bound organelles. So first we will discuss about the organelles without membrane. Some cell organelles like ribosomes and cytoskeletons are not bounded by any membrane. They are present in prokaryotic as well as eukaryotic organelles. So these organelles like ribosomes and cytoskeletons are common for prokaryotic cells as well as eukaryotic cell. Next, single membrane bound cell organelles. Some organelles are bounded by only a single membrane, thus they are called single membrane bounded cell organelle. For example, vacuole, lysosome, Golgi apparatus, endoplasmic reticulum, etc. They are present only in eukaryotic cell. So they belong to the eukaryotic cell only. Third category is double membrane bounded cell organelles. So cell organelles like mitochondria and chloroplasts, they are well developed or advanced cell organelles because they are bounded by the double membrane layer. So they are present only in eukaryotic cells. So they belong to the eukaryotic cell only. Our first cell organelle is cell wall. So cell wall is the outermost layer. It is a non-living, hard and rigid formation found in bacterial cell, fungal cells as well as in plant cells. This membrane is fully permeable, means it can allow any material freely inside or outside to the cell. It is made up of cellulose in plants. In bacteria it is made up of peptidoglycans and in fungus it is made up of chitin. So chitin is a complex carbohydrate. So in fungi the outermost layer which is the cell wall is made up of chitin. Function: it is a protective layer, so it provides the protection to the plant cell, bacterial cell as well as fungal cell. It provides rigidity because it is a very hard layer and it gives shape and turgidity to the organism. Our next cell organelle is cell membrane or plasma membrane. It is present in all cell, means it is common for prokaryotic as well as eukaryotic cell. just below the cell wall in plant cell. It remains present just below the cell wall, while it is an outermost membrane for all animal cell, because animal cell does not contain any cell wall. It is semi-permeable in nature, means it can allow the material in and out of the cell according to the requirement only. Okay, it is made up of bilipid layer with protein and carbohydrate. Thus, this model of the cell is known as fluid mosaic model, which was given first by Singer and Nicholson. Two scientists, Singer and Nicholson, describe this model. In your higher classes, you will learn further about this model, fluid mosaic model. In this model, in this diagram, you can see that this is the upper layer of the lipid and this is the lower layer of the lipid. In between, protein molecule, carbohydrate are present here. So this is called the fluid mosaic model, described or invented by the Singer and Nicholson. Functions. Cell membrane or plasma membrane encloses the content of the cell. It encloses the content whatever present inside the cell. It provides shape particularly to the animal cell, because in plant cell shape is provided by the cell wall, not by the plasma membrane. It allows the transport by the diffusion and osmosis. You must be knowing about the term, both the terms, diffusion and osmosis. It is the movement of any substance from its higher concentration to the lower concentration. Our next cell organelle is cytoplasm. Cytoplasm is a colloidal, viscous, jelly-like fluid remain present inside the cell. Here, this area is the cytoplasm. This is the cytoplasm. Cytoplasm contain 80 to 90% water and many organic and inorganic compounds present in it. It has various living cell inclusions called the cell organelles, means all cell organelles freely float inside this matrix, and as well as it has the non-living substances also, which are called as ergastic substances. Function. So cytoplasm contains enzymes responsible for all the metabolic activities taking place inside the cell. So it is a very important fluid present inside the cell. Next cell organelle is nucleus. Nucleus is also known as director or brain of the cell. Why it is called director or brain of the cell, we will get to know very soon. So nucleus is covered by a double membranous nuclear membrane in a eukaryotic cell. So nucleus is a prominent feature of a eukaryotic cell, because you already know that prokaryotic cell lack nucleus. So it contains DNA, RNA, protein, nucleolus and chromatin material. So means all our genetic material remain present inside the nucleus. So this structure is nucleus. Here you can clearly see that chromosomes are present here. Chromosomes are made up of DNA. So all the genetic material remain present in a eukaryotic cell inside the nucleus. Function. So why it is called director or brain of the cell? Because it controls all the metabolic activities of the cell. It initiates the cell division. It has a chromosomes or DNA which controls the heredity character. So it can transfer the character from one cell to another cell or one generation to another generation. So that is why it is responsible for hereditary character. Our next cell organelle is mitochondria. Mitochondria is called the powerhouse of the cell. It is called the powerhouse of the cell. It is a double membranous structure. Outer membrane is smooth. Inner membrane produce finger-like enfoldings called the cristae. Here in this image you can see that this is the outermost covering which is very smooth, and here in this innermost covering a finger-like foldings are present. These finger-like foldings are cristae. It is an autonomous body as it contains its own DNA. Thus it can self-duplicate. Here you can see easily inside the matrix of the mitochondria DNA is present. So it is the cell organelle which has its own DNA, so it is an autonomous cell organelle or autonomous body of the cell. This is the main site of the respiration. So function of the mitochondria is it can synthesize and stores the energy rich molecule ATP. ATP means adenosine triphosphate during the aerobic respiration. So that is why it is called the powerhouse of the cell, because it produces energy and it can store the energy in the form of ATP. Our next cell organelle is endoplasmic reticulum. Endoplasmic reticulum is called the framework of the cell. Why it is called framework of the cell? We will get to know this point very soon, but before then that I would like to tell you that endoplasmic reticulum is a network of membranes which extend from the cell membrane to the nuclear membrane. So it is a junction between the cell membrane and nuclear membrane. Here you can see that this is our nucleus and these are the endoplasmic reticulum. This area is endoplasmic reticulum. So it remains present from one side with the nuclear membrane and another side with the plasma membrane or cell membrane. There are two types of endoplasmic reticulum: rough endoplasmic reticulum, which is in short can be written as RER, and smooth endoplasmic reticulum SER. Why they are rough or smooth? Here you can see that this area is rough endoplasmic reticulum because small granules, these small granules, orange color granules which are known as ribosomes are present here, but at this area no ribosomes are present here. So this area is smooth in looking, so that is why it is called the smooth endoplasmic reticulum, while due to the presence of ribosome here this area is looking rough, so that is why it is called rough endoplasmic reticulum. Functions. So endoplasmic reticulum forms the skeleton framework of the cell. It transports the material from one cell to other. It provides a surface for the synthesis of materials like protein in rough endoplasmic reticulum and lipid in smooth endoplasmic reticulum. So smooth endoplasmic reticulum is responsible in the production of lipid, while rough endoplasmic reticulum is responsible for the production of protein. Formation of lysosome, Golgi bodies and vacuoles. This is also a very important function of the endoplasmic reticulum. Next, it helps in membrane biogenesis. What is membrane biogenesis? As you know that endoplasmic reticulum is able to form all the important constituent of a cell membrane or plasma membrane, which are protein as well as lipid. So it can form protein as well as lipid for the formation of plasma membrane. So thus it helps in the process of membrane biogenesis. Apart from this, it is also helpful in detoxification of harmful substance in the liver. Our next cell organelle is Golgi bodies. So Golgi bodies are known as, or Golgi apparatus are known as shipping and packaging department of cell. So these are the shipping and packaging department. Golgi bodies first discovered by the Camillo Golgi in 1898. It originates from rough endoplasmic reticulum. So the formation of Golgi apparatus is a function of rough endoplasmic reticulum. It contains sac-like cisternae and vesicle. It has two phases: cis phase, which is known as receiving phase, and trans phase, which is known as supplying phase. So in this image you can see that this area is called cis phase, from where it can receive anything from the other cell organelle, and after processing or packaging it can release the same material from this area, which is known as trans phase. So it has the two phases: cis phase, which is the receiving phase, and trans phase, which is the supplying phase. Function: it helps in the modification, packaging and transport of various materials. They store the product of endoplasmic reticulum. So it also works as a storage system of the cell. It synthesizes the lysosome and plasma membrane. So it helps in the synthesis of lysosome as well as plasma membrane. Our next cell organelle is plastids. Okay, plastids are the double membrane bounded cell organelle present in the plant cell, helps in the photosynthesis, and thus it is also present in photosynthetic or chemosynthetic bacteria as well as in euglena. These are of three types: chromoplast, different colored plastids, leucoplast, colorless plastids, and chloroplast, which are green colored plastids. So we should learn about the chloroplast, because chloroplast is known as the kitchen of the cell. Why it is called kitchen of the cell? Because it contains the green color pigment called chlorophyll, and you must be knowing that during the photosynthesis chlorophylls receive the sunlight which are essential for the photosynthesis. So it helps in the process of photosynthesis by which green plant make their food. Thus it is known as the kitchen of the cell. So matrix of the chloroplast is known as stroma. Stroma has membranous sacs called thylakoid, which are arranged one above the other to form granum. Each granum is connected by frets or grana lamina. Here you can see that this area, the liquid or jelly-like substance present inside the chloroplast is known as stroma. Inside the stroma, sac-like structures are present that are piled up one above another. So these are known as thylakoids. The stalk of thylakoid is known as grana. Each grana is connected by the frets. Functions: chloroplast perform photosynthesis, helps in the release of oxygen and formation of glucose or starch in the plant body. Chromoplast because it may vary in color, so it helps in imparting color to the flowers and fruits. Leucoplast, so being a colorless plastids, it only stores the various materials just like protein, fats and carbohydrates. Our next cell organelle is vacuole. So vacuoles are the single membrane bound sac-like vesicles, very small or absent in animal cell, while in all plant cells have the very large vacuoles. So this is the prominent feature of the plant cell. They have very large vacuoles in them. The membrane of vacuole is called tonoplast. In this diagram you can see that this is the outermost covering, which is tonoplast. Vacuole is filled with the watery fluid called cell sap, which has dissolved salts, sugar, enzyme, etc. Arise from endoplasmic reticulum and Golgi bodies. So Golgi bodies and endoplasmic reticulums are responsible in the formation of vacuoles in the plant cells. Functions: vacuoles can store cell sap, which may be liquid, solid or food particles or even toxic byproducts. It provides rigidity and turbidity to the plant cells. Next cell organelles are lysosomes. These are known as suicidal bags of the cell, natural scavengers, and even these are known as cellular housekeeper. Why they are named as so, we will get to know about this soon. So these are the single membrane bounded vesicles filled with hydrolytic or digestive enzyme, found only in animal cell. Lysosomes are usually remain absent in the plant cell. They are produced from Golgi bodies, Golgi apparatus. Functions of the lysosomes are: these are helpful in intercellular digestion of food in unicellular organisms. They can destroy the old and non-functional cells. They can recycle the worn out cells also. So in this diagram you can see easily this is a unicellular organism or it is a single cell. These are the lysosome. These small particles are lysosome. When a cell engulf a food particle into it, lysosomes by releasing the hydrolytic enzyme helps the cell to digest this food particle, and the remaining undigested material then excreted out by the cell. Next cell organelles are ribosomes. So ribosomes are called as the protein factories of the cell. These are the non-membranous cell organelles. They do not possess any cell membrane. They are present in cytoplasm, mitochondria, chloroplast, and also found attached with the rough endoplasmic reticulum and nuclear membrane. So they are located at many places inside the cell. So they are made up of ribosomal RNA, rRNA, and proteins. In prokaryotes, ribosomes are of 70S type, while in eukaryotes they have 80S type of ribosomes. What is the function of ribosome? Ribosome are the main sites of protein synthesis. So in this image you can see that this whole structure, this brown colored semicircular structure is known as ribosome. This is the small subunit of ribosome. This is the last subunit ribosome. So this image depicts the formation of amino acid chains which is a protein. So main function of ribosome is a protein synthesis. So after knowing so much about a cell, we can easily say that a cell is regarded as true biological atom. So in our next video, we will discuss another topic from the different chapter. Till then, stay home, stay safe, bye bye.